From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and concerned citizens making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Assistant Professor in Pollinator Health in the Department of Horticulture. The holy grail of restoration ecology for pollinators is to have a plant list that encompasses all the interactions between pollinators and the plant community that existed before disturbance. Now, it's easier said than done. You can look for bees and make these point observations of where bees are on flowers, but my next guest is going to make the case for a more comprehensive approach that combines both genetic techniques and also detailed observations of where plant communities are. Katie Arstingstall is a wildlife biologist working for NCASI, the National Council for Air and Stream Improvement, doing pollinator work here in Oregon. But before that, or earlier this summer, she had just completed her Master's of Science in Wildlife Science at Oregon State University, working with Dr. Sandy DeBano out in Hermiston. Katie's work revolved around this really wonderful technique known as DNA meta-barcoding, in which she would take pollen from bees and identify where they've traveled through the landscape. So in this episode, we're going to hear about that technique, but also she's going to tell us how it can be applied and specifically in the context of Eastern Oregon in better understanding bee communities, plant communities, and their interaction. That is going on all across the planet right now. the home office juggle. Now, listeners of the show know, I'm sure we've heard this before, that there's a tight connection between pollen, bee nutrition, and plant reproduction. But we've also all observed different bees being choosy about the kind of flowers they visit. To begin, can you tell us about some of the challenges that are associated with figuring out the pollen preferences of different bee species?
Yeah, sure. So I guess when we're out watching bees, visiting flowers, or trying to collect them with nets, we're limited in a few major ways. So typically as bee researchers, we're trying to describe interactions between bees and plants in pretty large areas and over pretty long time scales. But one person or even like a team of people can only cover so much area, and they usually have a pretty limited amount of time as well. So ideally, it'd be awesome if we could follow a bee from like the moment it leaves the hive or its nest to the moment that it returns after that full foraging bout. But that's really impossible because these bees can travel several miles from their nest or their hive. And so we're typically observing an area that's just like a few square meters when we're researching bees. So we end up observing just this really small snapshot of a bee's foraging behavior. So you could be watching a bee go from one lupin to the next and think, oh, this bee really loves
lupins. Yeah.
Yeah. But then who knows? I mean, the next second it flies 100 meters to a patch of bull thistle and goes to town over there. So, yeah, it's just really about being like limited, like physically and by your field of vision. It's really hard to get that full picture.
You know, I've often thought that there's, along those lines, that there's times of the year when researchers are out, and then there's like everybody's in class, and we don't really know what happens in the spring, which is a slightly different issue. But there may be the resolution of plant bee connections may vary for a lot of different factors. But if I understood you correctly, what you were saying was that if you're going out, you catch some bees and you see them on a specific flower, it may move around, and you're really just getting these very tiny snapshots. So we're kind of handicapped to a certain extent of being able to know all the flowers that they would visit in a foraging trip or over their entire lives.
Right, exactly.
Okay, on a previous episode, we had Pierre Lau from Texas A&M who talked about two methods that maybe could help with this, how to identify pollen on a bee's body, either using microscopic analysis or pollen DNA. And I guess if we could see the pollen on their bodies, it maybe give us a broader picture of where they've been. Can you remind us of some of the benefits and limitations of these different methods of analyzing pollen on a bee's body?
Yeah, totally. Microscopy is this method that a lot of pollinator scientists use, where you look at the pollen grains under the microscope and you can separate them out based on their physical differences to see what plant species they come from. And so this method is really cool because it helps resolve a lot of the issues with direct observation. So we're no longer really limited in time and space. All the pollen that bee collected is right there. So as long as we're able to identify those, then we really do get that like full picture. It's no longer really this snapshot anymore. But there are some limitations to this method as well. Microscopy is typically, as I understand it, you only use a subsample of the pollen load. So it is homogenized beforehand. So it should be a pretty good blend of what that bee was taking from, but you still could maybe miss some rare species that were in there.
So just to cover this, so you would get the bee, you would sort of knock the pollen off its body. You'd have to mix it somehow because the chamber for counting the pollen is only microliters in size. So you're only getting this small fraction, and you're making this assumption that you mix it up. But if there's just a little bit of pollen in there, the odds are you might miss it. That's what you mean by rare species might get missed.
Right, exactly.
Okay, gotcha.
Yep. And then another limitation is that the taxonomic resolution for this method is typically pretty low. So what I mean by this is that you're probably only going to get like a family or genus level identification as opposed to specific species. And the species level identification is really important when you're looking at plant bee associations. So yeah, from what I've understood from people who've used this method, you can only really ID pollen grains to that like higher taxonomic level. I've heard it's really, really difficult.
Yeah, it seems like there's a couple of very good specialists, but who are overwhelmed. But most people, it's a very challenging skill.
Mm-hmm.
Okay, what about this other method that you don't have to use a microscope and actually physically look at the structure of the pollen? How does that work?
Yeah, so in the past, I'd say like 5 years or so, it's become really popular. A lot of researchers have started using this technique. It's called DNA metabarcoding. So this allows you to take the pollen from the bee and sequence the DNA to we can use the DNA sequences to identify the species in a bee's pollen load. So instead of determining the species based on their physical appearance, you're using the actual DNA. And so I know Pierre explained how all plant species produce different pollen grains, and you separate them out based on their morphological differences. Well, similarly, we can tell species apart based on their DNA. And so I'm sure that you've heard, like as humans, we share 99% of our DNA with chimpanzees, and that 1% is what makes us different species.
Uh-huh.
And that's super similar with plants, in that they share the majority of their DNA as well, the different species, but there's a small amount of DNA that makes them different species. And so we can use these specific gene regions that are different between different species to separate out the plant species in a pollen grain. And so the way this works is you can take the pollen load off of an individual bee, and this might contain several different species of plants that it's collected from during that foraging bout. And we extract the DNA from the pollen. Then we use PCR, which stands for polymerase chain reaction, to amplify this specific gene region that is going to help us tell the different species of plants apart. And so then once we amplify these gene regions, we are able to download from the internet known DNA sequences for all kinds of plants. There's a ton available online. And then we can essentially just match the DNA sequences from our pollen samples to those of these known plant DNA sequences. And we can figure out what plant species are in the pollen loads from these bees.
This sounds super has much potentially more power than looking at the pollen grains under a microscope. Are there any downsides?
Sure, yeah. There are definitely some limitations to this method. One is there's a really big learning curve when it comes to understanding the different genetics lingo and learning all the techniques involved with the lab work, as well as there's a lot of coding involved, that was all new to me when I started this project.
Because there's so much data that you produce, you have to somehow congeal it.
Yeah.
Okay.
Yeah, 100%. And yeah, so it's a beast to get through all of the information. But another limitation of this method is it's not quantitative. So basically, we can tell what plant species are in a pollen sample, but we can't tell how many of each are in there.
Oh, and I guess coming back to your question of the person who wants to know which plants are most important for which bees, and you set it up real great at the beginning, is we only see them on one plant, but if at the end of the day you get all the pollen off them and it tells you the list, maybe they only kind of briefly visit. They don't really, that one plant's not that important, but it will look the same as all the plants because it'll come up as a kind of inventory of where they've been or something.
Right. Right. But there are some ways that we can kind of try to get around this. So for my project, we set up these transects in our sampling areas. And we basically would walk along these transects and count the number of blooming stems from each plant species that was growing along those transects.
Yeah.
So this gives us an idea of the presence and abundance of what's blooming in the area.
Yeah.
And so then you can use this data to essentially say, okay, well, let's say bull thistle, we only saw it 5% of the time on the transects, but we found it in 80% of the pollen loads. Then you could at least assume that that plant species is somewhat preferred by these bees.
Oh, because it's not there. There's not a lot of it out there, and it shows up on every bee as kind of probably trying to get to that one bull thistle.
Right, exactly.
And so that way okay, that makes a lot of sense. So although the tool may not give you the quantity, you can sort of infer it by walking the landscape and matching the two data sets together.
Yep.
Okay, fantastic. I have one more question about this before we take a break, then we'll dive into your research because I think we're already kind of dipping our toes in your research. But so these, is it? I'm not a genetics person at all, but you talked about regions of the DNA that you look for. Is this every plant has different regions, or there's one or two regions that you focus on? I'm thinking of it like a roadway. There's just mile 34 to mile 78 is where we're looking. Is that the case, or is it way more complicated than that?
That's a really good way to look at it. Yeah. It's essentially, so they have names, the two that I used in my project. One is called RBCL, and this is one found in the chloroplast DNA.
Okay.
So really good for plants. And then another gene region, we actually use 2 gene regions for my project because that actually gives you a little bit higher taxonomic resolution. You're able to identify more using 2 than 1 because sometimes plants might have the same RBCL region, but different ITS2 regions, which is the other gene region that we use, which is actually found in the nuclear DNA. So, yeah, they're just lots of people that are much smarter than me have done tons of research beforehand and figured out certain regions that are good for certain organisms. So there's some that are really good for plants. There's some that are really good for like fish. There's some that are really good for other organisms.
Okay, fantastic. Well, thanks so much. Let's take a quick break. I want to come back after the break and talk about your specific research, because it sounds like you've done this really wonderful kind of blend of, classical ecological work with this DNA work to be able to understand get at the central question that we raised at the beginning of, like, where do these bees actually go?
Yeah, definitely.
Okay. Okay, we're back. All right. So we were talking before the break. We were talking to the part before the break, how you've really tried to refine. You kind of made this bridge between these genetic methods and actual observation on the ground. Sort of refine these existing methods by first developing a library of plants that are actually growing in the area, rather than this huge library that you were describing on the internet where there can be plants from Madagascar and plants from England. You thought, well, one way I can get this whole system to work better is by really focusing on, what's the flora of that area. Can you tell us a little bit about how you went about doing that?
Yeah, definitely. So as I was mentioning when I was explaining the process of DNA metabarcoding, once you have your DNA sequenced from the pollen, you can match that DNA with what we call a reference library that contains known DNA sequences that people from all over have added to databases on the internet. And so we wanted to build a library that essentially just had plants in our area because certain families and genera can share of a very significant proportion of their DNA at these gene regions that we are using to identify species from. For example, 2 kinds of knapweed, spotted knapweed and diffuse knapweed, they differ in their ITS2 gene region, which is one of the ones that I used, by only 1 base pair out of 500. And so sometimes our pipeline can mix them up and can misidentify one as the other. And so when we were building this library, my advisor, Sandy DeBano, she has been working in my study areas for quite a few years, and she's been working with several botanists and other researchers at the Nature Conservancy and the Forest Service, and they've developed pretty comprehensive lists of the plants that grow in these areas. So we were like, oh, awesome. Like, let's just take these lists and kind of develop our library based on that. So when we were creating this, first we checked curated databases, which are essentially these like really top-notch databases where these sequences have been verified by botanists. So you can be like super sure that everything's very accurate. And so you can find the links to these kind of databases in different journal articles. That's where we found the ones that we used. And then everything that we could not find from those curated databases, we downloaded from an open source database called GenBank, which essentially anyone can sequence DNA and add it to GenBank. It has to get accepted and go through an application process, but
you had two tiers. You wanted to go with the really verified sequence data first, and then if there was something missing. Can I just ask you a quick question on this? So I imagine, the amount of plants that bees visit in the study area you're working were hundreds. So you had to go through line by line. It's like, this plant, where's the best sequence? And then kind of that sounds like a lot of work.
Yes. So that's where the coding comes in.
Okay.
And so I had this really awesome colleague. His name's Xiaoping Li. He's a postdoc with Ken Frost, who's in the Department of Botany and Plant Pathology at OSU. So Xiaoping actually wrote up a little script that he basically could just run it, and it would essentially check what was in the database and give you back ones and zeros if it was there or not there. So yes, by hand that would have taken
forever. Okay, so you started with this great data set of actual plants on the ground, and then you had to assemble a corresponding database of sequences using this fancy super program. And then what? And then how did you proceed from there?
So I guess on average, we were able to download about 70% of the DNA sequences for about 70% of the plants that were in our plant lists.
Okay.
Which is a pretty good amount. And so then essentially we run another pipeline to match those sequences. And then, like you were saying before, it essentially spits out a list of what plant species were found in each pollen load. And so but to compare, one thing we wanted to do was to compare this data to actual behavioral observations, like we were talking about in the beginning. So as we were catching these bees, we were noting the flowers that they were visiting while we were sampling them. So we can essentially directly compare observation data to our metabarcoding data to see if there were any differences.
Ooh. That sounds exciting. Okay, and what was the answer? Because I guess this was how you've set things up. Is this is the way we've been doing it for a long time. When we say, oh, this bee's a specialist bee, it's maybe based on a couple of observations of the bee on a flower. So when you lined up the proof in the pudding, the actual the genetics with the observational data, what did you see? Was there a lot of were they visiting a lot more plants than we might have thought they were?
Yes, they were. So that basically we found that their diets are a lot more complex than what you would assume based on just direct observations. For example, at Starkey Experimental Forest and Range was one of my field sites out in Eastern Oregon. We documented 26 plant species that bees were observed visiting. But when we looked at the metabarcoding data, we actually found 44 plant species in those pollen loads, so almost twice as many species. And we also, not only were we detecting more species, we were detecting more interactions per species. So each bee's pollen load contained anywhere from 1 to 9 different plant species. And so, like you said, a lot of these bees that appeared to be specialists based on just observation now looked like they had more of a generalist diet. So it's super interesting.
Well, and I think that it's also just the first point as well, that when we go out and we do these surveys of plants and the bees they visit, we may be underestimating by a lot the number of species that are actually being visited. So when you mentioned earlier this kind of, going through and seeing, what plants were actually out there. Was anything surprising? Were there plants that were sort of performing much differently than you would have thought, given just doing observational work?
Yeah, definitely. One in particular that I think of is yarrow. At least in my experience, I don't see bees on yarrow very often.
Yeah.
But it was like top. It was probably like the third most common species in the pollen loads at 2 of our locations.
Wow.
Which was really cool. Another one that comes to mind is so I had some field sites at a farm, a large industrial farm. We sampled in the field margins, and there was a lot of rabbit brush, just not in our plots. So we randomly selected, I think 8 field margins to sample from. And so there was a lot of rabbit brush in the surrounding area, just not where we were sampling in. And so I had no observations of bees visiting that species. But it was the second most common species in the pollen samples. So, I mean, that was like a perfect example of like, I mean, we never would have known that they were even using that if we had not done the metabarcoding.
I love, the thing that you said at the beginning, like most researchers work on a plot, and like bees don't work on a plot. And so you may be so focused on, because you may be doing some kind of restoration on a plot, and you're really focused on it, but your bees may actually be like, they're hanging out in the plot, but they're doing a lot outside the plot.
Right.
You never know. Yeah.
Yeah. It was a lot of fun looking at the data, because it's like, I don't know, like I also remember elephant's head being in the pollen, which is that really cool, like spike hot pink flower that looks like an elephant head. And I remember seeing that at Starkey like one time, but that was showing up in there. Just lots of cool stuff that I never would have even known that they were using.
Well, why don't you kind of take us through a little bit, because it's a part of the state that I think has some remarkable bee biodiversity, and many of our listeners may have not been out there. Tell us about some of the other cool plants that popped up through your analysis, and some of the bees that you ran into and caught.
Yeah, definitely. So I had 3 different study locations, all in Eastern Oregon. They were all relatively similar to each other, but very different ecosystems. So I worked at the Zumwalt Prairie Preserve, which is this remnant bunchgrass prairie. It has like a very rich forb community. Has a lot of flowers that are typically found at higher elevations, like clarkias, paintbrushes. There were some larkspurs, lots of lupin. And the bee communities out there were pretty diverse. We sampled about 35 species from Zumwalt. I would say Zumwalt's pretty dominated by bumblebees, or at least that was most of what we caught. Specifically, we had a lot of Bombus californicus, Bombus centralis. And then we had a mix of all the other genera as well, lots of Andrena, Halictus, Lasioglossum. But the bees at Zumwalt definitely loved the lupin, and that's been my experience like everywhere I go typically, is if there's lupin, the bees are going to find it. At least the bumblebees love it. And then my second field site was at Starkey Experimental Forest, which I had several forested riparian sites there. So my sites ran along Meadow Creek, which is a tributary of the upper Grand Ronde River. And so there's also a high diversity of flowering plants and bees at Starkey as well. As far as plants, the Zumwalt and Starkey overlap quite a bit, but at Starkey you would see more Missouri goldenrod, St. John's wort, penstemons also really common. And we actually sampled individuals from 40 different bee species there. Again, a lot of bumblebees at Starkey. We collected a ton of Bombus bifarius. We also caught quite a few Halictus lasioglossom species as well. And then my last site, 3 Mile Canyon Farms, which is, I would say, a pretty disturbed landscape.
This is in Eastern Oregon, but whereabouts? I'm trying to...
It's right next to the Boardman Preserve. It's in Morrow County.
Okay.
Yeah, really close to Hermiston, where OSU has their extension center. It's a pivot irrigated farm. They keep their field margins uncultivated.
Oh, you know, I've been there. It's that place that does all the international hay exports.
Sure. Yeah, I didn't know that, but
okay. But so
they do a lot.
But that was also the site that you described earlier when you were talking about the plots and the rabbit brush.
Yes.
Okay.
Yes.
All right. How did the bees look out there?
Very different. The bee communities and the plant communities were very different at 3 Mile. As far as plants, lots of non-native vegetation. There were several knapweeds and thistles like Russian thistle and Scotch thistle. And as far as bees, less diverse as far as what we sampled. We sampled 24 bee species, but I was still I was very surprised at how diverse it was there, because when I first started sampling there, I was like, oh, it's going to be all honeybees. I'm not going to find any native bees, but there were a lot, and super pretty ones, like lots of Aegopostamon, the green metallic bees, lots of digger bees. They love that soil. Mm-hmm. Several Melisode species. I'd say a lot less bumblebees at 3 Mile.
You know, it's something that I've noticed, working in the Dalles this summer, and there is a way that just after you leave Hood River and you head, to Hermiston, it's just not a great bumblebee area. But then you go up in elevation to Starkey and Zumwalt, then it suddenly is. But it just strikes me that whole stretch is just not, I don't know, it's too dry, it's too early, there's not enough late bloom or something. But it really is. I've noticed the same thing.
Yeah. Yeah, it's kind of like a desert there for a while. Mm-hmm.
Well, those are 3 really different sites, so it must have been a really good confirmation of this approach, because you would have had, if you were able to have very good correspondence, a better correspondence between your genetic and between your genetic data and the actual plant community, that would be a good test of it. You would be looking at a real broad range of landscapes.
Yeah, definitely. And we haven't started writing this paper yet, but we are going to look at several factors comparing the bee communities and the plant communities between those very different landscape types. And what we found is that it seemed like the bees at 3 Mile were exhibiting a lot more of a generalist diet. So it's almost like they're just making do with what they have out there, using a little bit of everything. Whereas at Starkey and Zumwalt, where we have this high plant diversity, there's a lot more room for specialization, is what it looks like.
Oh, that would be a really great finding to document. I think that's really interesting. Well, let's take a quick break. We'll come back. We have our questions that we ask don't ask our listeners. We ask our guests. But let's take a quick break and we'll come back.
Sounds good.
Okay, we're back. So first question is, do you have a book recommendation for our listeners?
Yeah, so I can almost guarantee somebody else has recommended this, but it's so good. The Bees in Your Backyard is my favorite. Just the pictures are amazing, but it's also full of so much awesome information about native solitary bees. Goes through all the genera specifically, so I really love that book.
I remember seeing it for the first time at the University of Calgary. Lincoln Best showed up with a suitcase of them, and he said, "You guys all need this." But the other thing I do know is that we were talking to Olivia Messenger-Carroll, one of the co-authors, and I think they've sent to the publisher the bees of eastern U.S., and they're working on bees of western U.S. So same really great images and lots more detail. I'm so excited to see those.
That's awesome.
Great recommendations. Also my favorite, too. So I imagine you do a lot of different things. You've had to master a lot of like esoteric techniques and technologies. So I'm really curious what your question is going to be to your answer is going to be to this question. What is your go-to tool for the kind of work you do?
Yeah, so I had to think about this one because my tools are pretty spread out. But one thing that is so important in identifying plant-pollinator relationships is plant ID. If you don't know what you're looking at out in the field, you're going to have you're not going to get as great of data as you want. So I picked the Wildflowers of the Pacific Northwest. It's a field guide by Mark Turner and Phyllis Gustafson. It's organized by color and then by shape. So it'll have like yellow flowers, and then it'll say like 3 petals, 4 petals, 5 petals. And so it's super like user friendly. If you don't have even if you don't have any experience in plant ID, it's really great. It's got awesome pictures.
Fantastic. We'll put a link to that in the show notes. I recently, there's a really, it's really vulgar, but there's a really great YouTube channel called Crime Pays, Botany Doesn't. And but the one thing that so the guy does this. He's some kind of like train conductor from the Bay Area who travels all across the West, and he does botany. But he did emphasize to me the importance, because I'm the same way. Like I like the Oregon Flora guides, the app with the flower shape and then the petal colors. But he really did emphasize to me, getting a basic book on basic botany and taxonomy, like how the plants are organized. I've never really done that. I've never, I know these plants are related, but I imagine you've had to do that a lot. That you now have a really intricate knowledge of how these plants are related. Because one could be pink and one could be white, but they're just basically the same plant. I'm turned on to that right now. I'm going to try and crib this winter on plain systematics of plants. Yeah. Okay, well, that's a great recommendation. I'm always looking for a great field guide. It'll be in the show notes. And the last question we have, it's always an unfair question for somebody who's probably has a lot of different pollinators that they love. But is there one in particular when you see it fly by, you're like, I love that thing?
Yeah, I do have one. The scientific name is Anthophora curta. The common name is short sun digger. It's a digger bee. It's just so cute. It looks like a little teddy bear. It's got these like teal eyes and like light blue hair. I just think it's adorable. Really furry legs. I love it.
So it's Anthophora. What was the species?
Curta.
Curta. Okay.
Curta. And
so it's not one. It's not an Oregon Anthophora, is it?
I'm not sure. It was all over at 3 Mile Canyon Farms.
Oh, it is. Okay. I never seen a bluish one. That's really cool.
Well, yeah. I mean, it's not like blue, but it's just like light and awesome.
Fantastic suggestion. I am looking for. It would be really great to have a blue Anthophora. I'm gonna now hunt for the blue Anthophora.
And they're super hard to catch. They're extremely fast. They're like buzzing all around.
I remember trying on Larkspur. I remember seeing them, and it was just like you had to sit for a long time and plan your collection because you were never going to catch it. It's sort of like, it's going to come back in 3 minutes. I'm going to wait.
Exactly.
Well, this is fantastic. Thank you so much. I think this has really kind of filled out our understanding of these complicated connections between bees and pollen. And good luck with writing up those papers. We're really excited to. Maybe we'll have you back after it's published so we can discuss them.
Yeah, that'd be great. Thanks for having me.
Thank you so much for listening. The show is produced by Quinn, Sin, and Neil, who's a student here at OSU in the New Media Communications program, and the show wouldn't even be possible without the support of the Oregon Legislature, the Foundation for Food and Agricultural Research, and Western SARE. Show notes with links mentioned on each episode are available on the website, which is at pollination.podcast.oregonstate.edu. I also love hearing from you, and there's several ways to connect with me. The first one is you can visit the website and leave an episode-specific comment. You can suggest a future guest or topic, or ask a question that could be featured in a future episode. But you can do the same things on Twitter, Instagram, or Facebook by visiting the Oregon Bee Project. Thanks so much for listening, and see you next week.